and then we will get started. So on Tuesday, we looked at the metabolism of other sugars and how they find themselves going into metabolism. Then we started talking about the pentose phosphate pathway, and we stopped here with just an overall view of the pentose phosphate pathway. So today we're going to go into depth of how this works, why this pathway exists, and then we're going to move on, look at the big picture, how it connects with glycolysis, and then move on to glycogen. Right. So let's actually look at this pathway step by step. And it's broken down into 3 stages. Oops, I forgot to turn off the waiting room. There we go. So it's broken down into 3 different stages. And let me do some rewriting here for what's cut off at the bottom. They're just molecule names. So we have glucose 6-phosphate. This is 6-phosphoglucono delta lactone. This is 6-phosphogluconate. And this is ribulose 5-phosphate, also known as RU5P. All right, so to begin with, we start in this pathway with G6P. G6P is, if you remember, the first step of glycolysis. We take glucose and we make glucose 6-phosphate. So hexokinase can make G6P, or as we're going to look probably starting next week when we talk more about glycogen, or at the end of today, we can see we can actually get glucose 6-phosphate if we break down glycogen. So the first step of this pathway is to take this glucose 6-phosphate and make it into a lactone. And when doing this, we make NADPH. All right. So what's going on here is that we're taking 2 hydrogens from glucose 6-phosphate, so this hydrogen and this hydrogen, and we're using that to make NADPH. I mentioned this on Tuesday, but the way these arrows are made, these 3 arrows, that's just showing you this is not reversible. This just goes in one direction. This NADPH, also mentioned on Tuesday, but just as a reminder, is used for reductive biosynthesis. So this molecule is just mainly used to make other things in your body. And this reaction strongly inhibited by NADPH, which makes sense, right? Since step 1 is irreversible and we can use glucose 6-phosphate for other things like glycolysis, we don't want to use this pathway if we don't need NADPH, right? The first step is already committed. Once we do the first step, there's no turning back from this pathway. So this is more or less product inhibition or feedback. So product inhibition. You'll also see this as like feedback, product feedback, feedback inhibition, where the thing we make goes and actually turns off the enzyme that's making it. So from there, we make 6-phosphoglucolactone. What a lactone is simply the group that I'm highlighting here. It's this oxygen in a bond and then C double bond O. That's our lactone. Once we have our lactone, what we're going to do is we're going to hydrolyze it to form 6-phosphogluconate. So if we look at what we're doing here, we have this ring structure, and we have this double bond oxygen. So we're going to actually add an H to that oxygen. Actually, that's not the right one, but we're going to add a H to that oxygen and then H to the carbon as well to form our 6-phosphogluconate. And here we're showing the sugar in the linear structure. Remember, although we show glucose, and this is from Biochem 1, although we show glucose in the ring structure, these rings can open up and form the linear structure, and that happens all the time. It's in a balance between being in a ring and being in linear. So we're just showing it in linear form here to better show what's happening. The next step is that we take 6-phosphogluconate and we're going to run it through a dehydrogenase. Remember, when we see the word dehydrogenase, I've been hammering this all semester, you know you're doing a redox reaction. Here, we're making another NADPH, and we're also chopping off a CO2. So we're taking our 6 carbon sugar, and making it a 5-carbon sugar, ribulose 5-phosphate. That's why this is called the pentose phosphate pathway. The pentose comes from 5 sugars. So that's the number 5. The phosphate is because we have a phosphate along those sugars, so pentose phosphate pathway. So for every 1 molecule of G6P that goes in, we get 2 molecules of NADPH. Remember, NAD or NADP, whenever you see that, electron carriers, don't forget about that. If it has the hydrogen, it already has the electrons on it. We can then use those molecules to do other reactions. And just as a further reminder, NADPH and NADH are different. They're not equal, and you cannot interconvert between the two, and they are used for different reactions. So don't forget about that, just in general. So that's the end of stage 1. And once that's done, we can go on to our second stage and use our ribulose 5-phosphate. Dehydrogenase is an enzyme that does redox reactions. So normally when you see dehydrogenase, what that means is that it's going to use NADH, NADPH, or FADH2. Usually you're going to see one of those. So that's your cue for redox reaction. Any other questions about information presented here on stage 1? All right. So stage 2 is that we take our ribulose 5-phosphate and we're going to convert it into 2 different things. We're either going to convert it into ribose 5-phosphate, R5P, or we're going to convert it into xylulose 5-phosphate, XU5P, or XU5P. If you look on the image here, the arrows are actually telling you the relative amounts. 33% of the time, usually, we go this pathway. 66% of the time, we go this pathway, or a 2-to-1 ratio. For every 1 molecule of ribose 5-phosphate you make 2 molecules of cellulose 5-phosphate. That can change. That can, of course, be changed by enzyme amounts or different allosteric modulators, but in general, that's what's going on. So to do our first reaction, going from ribulose to a ribose, that's an isomerase. Remember, an isomerase, all that's doing is it's just kind of like changing, more or less bond ordering. The easiest way to think about it here for this reaction is that you're just flipping where the carb C double bond O is. So on ribulose 5-phosphate, C double bond O is on carbon 2. On ribose 5-phosphate, it's on carbon 1. Otherwise, the molecules are identical. That's all that's happening. In our ribulose 5-phosphate epimerase, we're doing an we're creating the epimer of ribulose. Hopefully you remember from Biochem 1 and what we talked about earlier in this semester. Excuse me. An epimer is simply where you have one OH flipped. And by flipped, I mean what side are you on in the linear form. Here, we're flipping carbon 3. So usually in ribulose, they're both on the right. In xylulose, carbon 3 is now on the left. So for this pathway, it's important to make NADPH. That's one important thing to make. So talking about the pathway in general, just not stage 2, one of the important products is NADPH. The second important product is ribose 5-phosphate, because this ribose is going to make nucleotides, right? DNA, RNA are either ribose or deoxyribose. This is how you get the ribose in your body. It comes from sugar. Kind of wild to think about, but it makes a lot of sense, right? Your DNA has sugar residues. How do you make those? Well, you eat stuff. So you take that glucose and doing this pathway, you make that. In rapidly dividing cells, you might make a lot of R5P. Can anyone know of example of a rapidly dividing cell where you might get a lot of R5P? In step 5, are we creating the epimer of ribose? Your key in step 5, you are creating the epimer of ribulose. So step 5, you're creating the epimer of ribulose. Cancer cells, correct. So in cancer cells, this is a pathway that will be utilized a lot because cancer cells rapidly divide. So they need a lot of nucleotides in a short amount of time. So they're going to be utilizing this pentose 5 pathway quite a bit to make those new nucleotides. And also that NADPH can be used to make stuff as well. So is there any questions so far about pentose phosphate or what's happening in stage 2? Okay. Stage 3 is easily the most confusing stage when you look at it, at least. But we're going to go over it, and I think this image on the right color codes things quite well to show what's happening. But just so we're oriented, on the previous slide, we ended here, and we made these 2 molecules. So that's where we're picking up. Now, we're going to assume when we go over the stage 3, we're going to say 3 molecules of glucose 6-phosphate have already entered stage 1 and stage 2, right? So we had 3 glucose 6-phosphates come in. We made 3 ribulose 5-phosphates. And then in stage 2, we currently have 1 ribose and 2 xylulose 5-phosphates waiting for stage 3. Okay, so the idea of stage 3 is that we're going to take these 2 5-carbon compounds and we're going to change them into 2 6-carbon compounds and a 3-carbon compound. So it's cut. The very last thing on the bottom left is cut off. This says sum. Whoops. Our sum reaction is that we take 3, I might have said 2, but 3 carbon 5 compounds. We're going to make 2 carbon 6 compounds plus 1 carbon 3 compound. So we start with 15 carbons. We end with 15 carbons. And we're just doing some fancy reactions to mess around with our skeletal structure here. So we're going to start with 6. Ignore everything that's happening at the bottom right now and just look at 6. The 6 is a transketolase, right? So what's happening in this transketolase is that you take carbon 1 and carbon 2 from xylulose, and you just attach them to the top of ribose 5-phosphate. That's all you're doing. You're plucking 2 carbons from one compound to put them on another compound. And what you're making is cetohepulose 7-phosphate. Those of you, that's a really confusing name. It makes sense once you understand like hepulose, sugar 7, hep. S7P is a lot easier to remember, especially because the S, you can make it stand for 7, right? 7. So you're going to make a 7-carbon compound with a phosphate on the 7th position, S7P, plus glyceraldehyde 3-phosphate, GAP. Where have we seen GAP before? This isn't the first time we've encountered GAP. Where else have we seen that? You want to remember where GAP comes from or where we've seen it this semester? Glycolysis, correct. GAP is at the end of like step 4. This is what GAP is, the molecule that feeds into stage 2 of glycolysis. So this, we've seen this before. It can be fed into stage 2 of glycolysis. This idea of seeing multiple sugars or the same sugars in multiple pathways is going to be something that occurs again. Because remember, although we're looking at these pathways all individually, everything's connected in the cell. All these things are happening at the same time. So it's all very interconnected. So our step 6, and I have it here, we're taking a C5 plus a C5, and we're making a 7-carbon compound and a 3-carbon compound, right? Then we're going to step 7, which is a transadolase. So what we're doing there is we're going to take the top 3 carbons from our cetohepulose, and we're going to attach them. going to just erase this. We're going to attach them to the top of GAP. When we do that, we make our friend fructose 6-phosphate, which is also used in glycolysis. And what's left from our acetahepulose is just a 4-carbon compound with a phosphate on it. This is E4P, or etherose 4-phosphate. So E4P is easier to say for sure. So stage 7, we are taking a 3-carbon compound plus a 7-carbon compound and making a 6-carbon compound and a 4-carbon compound. All right. So for fructose, this is finished. The fructose 6-phosphate, it can go reverse on step 7, but it is not used in step 8. That's what I mean by finished. Not used in step 8. So if fructose 6-phosphate wants to, it can go back to glycolysis. Or it can go reverse here, but is not used further on. What is used is our E4P, right? So I'm going to go back to our end of phase 2 box I have. And up to step 7, I have used my ribose. So I don't have any ribose left. I used the one already in step 6. I use one of my xylulose in step 6, but I still have one left. That's what's going on in step 8, and that's what this arrow is showing as well. In step 8, we're taking xylulose 5-phosphate, a different molecule of xylulose 5-phosphate, and we're taking etherose 4-phosphate, and it's the same reaction. In fact, the enzyme types are the very same. They're both transketolases in step 6 and step 8, and the reaction is 100% the same. We're taking the first 2 carbons from xylulose, and this time we're attaching them onto the top 2 carbons of etherose. And what we make is another molecule of fructose 6-phosphate plus GAP. So if we end at step 8, our products from the pentose phosphate pathway, all in all, we made 2 molecules of fructose 6-phosphate, 1 molecule of glyceraldehyde phosphate, or GAP. And we have completely used up our ribose 5-phosphate and our xylulose 5-phosphate. And like I said, this is all reversible. That you can tell from the double arrows, all of this is reversible. So depending on what the body needs, you can go in any direction. What I mean by that, if you want to create more nucleotides, what you can do is that you can take GAP and fructose 6-phosphate from glycolysis and run this pathway in reverse. Like you start at step 8 and you go to 7 to 6 to make more ribose. So although this is stage 3, keep in mind that fructose and GAP are both part of glycolysis. And so you can start the pathways from there and remake ribose 5-phosphate. Or you could do what we just showed in the series of 3 slides, start from glucose 6-phosphate, work your way down here to this stage as well. Any questions about that? or any information you might want me to repeat or anything at all. All right. If not, we can move on then. Hopefully that was a clear explanation of what's going on. So let's take a break from the pathways and just do some self-tests to make sure we kind of understand, and some questions to get us thinking about the bigger picture, because we've been zoned in on all these pathways, but I don't want to lose focus of how this impacts the cell. So the first question I have here, and one I have not talked about at all yet: why do you think the pentose phosphate pathway happens in the cytosol of the cell? Is it because glycolysis also occurs there? 100% correct. We just talked about, right? Pentose phosphate pathway starts in one of two places. At stage 1, we start at glucose 6-phosphate. Sorry, I really need to work on my G's and my 6's, because I just wrote 6G phosphate, but glucose 6-phosphate. Stage 3, we start with fructose 6-phosphate plus glyceraldehyde phosphate. Either one of those we can start at, depending on what we want to make. We start at stage 1 when we want to make NADPH for the most part. We start at stage 3 when we want to make ribose 5-phosphate for the most part. Stage 1 can also make ribose 5-phosphate though as well. So it can also make that. Stage 3 cannot, let me just make that clear, cannot make NADPH. So we start at stage 3, we cannot go back to stage 1. But everything's connected. So to start this pathway, I need to be fed in these sugars. These sugars are created from glycolysis. Glycolysis happens in the cytosol. Therefore, it makes sense that all the phosphate pentose pathway enzymes are also in the cytosol, because they are working on the same things. Now we'll see later in this class some of these cellular locations. They are different, and we need special proteins, special enzymes to transport stuff across like membrane barriers. Here we don't. It's all in the same place, so that makes it easy for us. So yes, it's because it's where glycolysis takes place. So here's another question, one I have a pull for. And hopefully you're paying attention, so hopefully this one's kind of easy for you. Let me get that up. Which of the following are products of the oxidative phase of the pentose phosphate pathway? What are we making in the oxidative phase? It would be best if you don't look at your notes for this to try and remember, but if you're drawing a 100% complete blank, feel free to go look at your notes. But this is another way to test yourself, strengthen your brain. All right. Seems the majority of us got it, so very good. So it is E, not D, E. So just when going over this, again, some ways to tackle these multiple choice questions. Hopefully, I've hammered away that pentose phosphate pathway uses NADPH. So no matter what, we can eliminate the first three because it's not NADH. Now, the oxidative phase, the only phase that does redox reactions are phase 1. So that's our first 3 reactions where we go from glucose 6-phosphate to our lactone. Then we go to our ribulose. So we're not making ribose. Plus, another way to really think about this is that we're going from a 6-carbon sugar to a 5-carbon sugar in step 1 because it's pentose. We need a 5 sugar, pentose. Remember, ose, every time you see ose, that means sugar. So another strategy to think about how to answer this on multiple choice is you're losing 1 carbon. Here you're losing 1 carbon. Here you're losing 2 carbons. So if you don't remember if it's ribose or ribulose, because those words are very similar, hopefully that strategy showed you like how to tackle this. And it's ribulose 5-phosphate, 2 NADPH, and 1 carbon dioxide. So that's from stage 1, the pentose phosphate pathway. So question 3 is another big thinking, critical thinking question. Cancer cells can have a different isozyme. An isozyme, if you remember, is an enzyme that does the same reaction, but they're actually like different. So they have a different isozyme of pyruvate kinase. This isozyme has lower activity compared to pyruvate kinase in normal tissues or other tissues. What does this mean for cancer cells? And how can it promote the growth of cancer cells? So based on everything we've talked about this semester, when it comes to glycolysis and now the pentose phosphate pathway, take a few minutes, think about this. If you have a suggestion, an idea, please feel free to share it to me. But otherwise, I'll give you some time to think about this one. So if it's inhibiting a checkpoint by having lower activity, would growth go unchecked? So I don't know if it's so much that growth will go unchecked because you are inhibiting. Well, I'm trying to formulate my answer in the best way possible. So you are correct in that we are more or less not inhibiting, but slowing down pyruvate kinase. That's what lower activity means. It's not going, the flux is slowed down. But pyruvate kinase is not an important checkpoint for glycolysis. Remember, pyruvate kinase is the very last step, step 10. So it is far from equilibrium, but it's not as important as step 3. So you're on the right lines. I'll just look for you to flesh out why that would create growth. Does it favor increase of the pentose phosphate pathway and the production of precursors for nucleotides for DNA synthesis? it does. Why? Why does it do that? You're correct. I want you to take that extra, again, take that extra step forward and explain why inhibiting or slowing down the last step of glycolysis would increase the pentose phosphate pathway, increase the rate of the pentose phosphate pathway, which we need for growth in nucleotides. So both of you so far have been correct. Marry those two pieces of information together into one big theory. Pyruvate is not an inhibitor of the pentose pathway. Good idea, but as far as I know, it is not. What happens? Let's think about this idea, right? Let's say in glycolysis, the steps that really matter for flux are step 1, step 3, and step 10. Right? Step 10 is our pyruvate kinase. And step 3 is our enzyme that really dictates flux. And remember, when this is on, normally what step 3 can do, the product of step 3 can go on and turn on step 10, pyruvate kinase. So that's normally what's happening. But in cancer cells, the activity of pyruvate kinase is way down. So remember, so step 3 in glycolysis is more or less irreversible. But what happens if step 10 is slowed down? What happens to all the intermediates here? Do the intermediates of glycolysis, and by intermediates, I mean anything that's not pyruvate or glucose, do the intermediate concentrations go up or do they go down? It's not so much they speed up. So I guess I should be more clear. I'm talking about the concentrations of our metabolites. So like glyceraldehyde phosphate or fructose 6-phosphate. But those intermediate concentrations would go up, right? Because normally in glycolysis, what happens is that pyruvate kinase takes, the molecule, makes it pyruvate, pyruvate leaves to do something else, and it pulls the whole flux forward. It helps to pull the flux forward. So if this is slowed down, you would have this buildup of intermediates. And we just showed some of these intermediates, like glyceraldehyde phosphate and fructose 6-phosphate, and to another extent, glucose 6-phosphate, they go to the pentose phosphate pathway. And the phosphate pathway. In the phosphate pentose pathway, if we want to build stuff, we're either going to make NADPH from glucose 6-phosphate, or we're going to make our ribose 5-phosphate. This makes nucleotides. And this can do redox reactions to make stuff, make other molecules. So the idea here, and it's actually quite clever, I mean, cells aren't thinking, so it's just a random mutation, but it's a really lucky mutation because what happens is that you increase the concentration of things like glyceraldehyde phosphate and fructose 6-phosphate, and to a lesser extent, glucose 6-phosphate. Then you take these away into the pentose phosphate pathway. So when you take them away, their concentration will then drop. And if you take these away into a separate pathway, you're not using them to make ATP. So overall, the amount of ATP will slowly decrease in the cell, and the amount of AMP will increase. Remember, ATP turns into AMP after it's used up, or ADP. With an increase of AMP, and there's multiple things happening here. So let me continue with the AMP story. AMP increases. This goes and turns on the enzyme in step 3. Remember, we talked about that, I think on Tuesday or last week, which is the main control point for flux and glycolysis. So we're going to keep making more glyceraldehyde phosphate and fructose 6-phosphate to be fed into the pentose phosphate pathway. And since these two metabolites are also being fed into the pathway, if you remember Le Chatelier's principle, what that says, and sorry, I don't speak French, so I always butcher his name. That says that if a reaction is in equilibrium, and so you have a reactant and a product, and let's say I take product away from this reaction, what that principle says is that the forward reaction will speed up to make up for that product being taken away. Well, I'm taking away GAP and F6P, so glycolysis will, the enzymes of glycolysis will make more GAP and F6P, which can be then fed into the pentose phosphate pathway to make more stuff. And also because you are, using these metabolites for different things, the cancer cell is going to need more and more glucose to both make energy through glycolysis and make new molecules through the pentose phosphate pathway. So they're going to steal glucose from their host body. I say host, but cancer cells are you. So they're just going to steal resources, which is what you see for cancer cells, right? They just gobble up all the glucose available. And this is one of the reasons why they do it. They slow down pyruvate kinase. They throw flux out of balance. Metabolites build up. Therefore, these metabolites that are building up will be used by different enzymes. Since this flux is slowed down, your ATP is going down. So these cancer cells are always sending out signals of, I'm hungry, I'm low energy, give me more glucose. And so your body delivers them more glucose at the detriment to your other cells. So that's kind of like the big picture, how even though we're looking at everything minutely, don't forget this is all tied together. And by inhibiting one enzyme of a separate pathway, you are influencing another pathway here in pentose phosphate pathway. What would happen if we did something to stop the signals that said we need more ATP? You would probably kill the cell. Like if you were not creating ATP and your body did not know that, or the cell didn't know that, then the cell would die. But that's kind of hard to do. In the cancer scenario, yeah, the cancer cell would probably die. That's hard to do because remember the signal for being low energy is AMP. So ATP, right, makes AMP by getting rid of its phosphates. And that is the signal of saying I'm low energy when you have a lot of that around. So it's if for some reason that all and AMP is recognized by like hundreds of enzymes. But if for some reason none of those enzymes recognize that, the cell would die because it would not speed up making ATP. Yeah. So that is one way to kill a cell, but it seems very hard to do to put that into practice. And I mean, that's the problem with cancer cells in general, is that they're your cells. So to really kill them, your own cells are going to be hit as well. But I mean, one way to know if you do have cancer cells and where they're located is you just see what cells are taking up a ton of glucose in your body, because that's what cancer cells do. But any questions about like what this is saying, like this question, or how we're going to talk about a little bit more how everything's connected, but anyone have questions on top of their head right now? And I never said it, but I probably should have said it. Instead of saying step 3, remember that's phosphofructokinase, PFK, is the enzyme in step 3. I was just being a little lazy and saying step 3 there. And step 1 is hexokinase. That's the enzymes that are responsible for step 1, 3, and 10. So basically the answer to this is that the intermediates build up, which will be fed into the phosphate pentose pathway, which is used to make nucleotides, and also NADPH is used up or is made, and that can be used to make other molecules we haven't talked about yet. Where does ATP go since it goes down? ATP is used for everything inside the cell. I know that's a blanket answer, but making proteins, you need ATP. Making nucleotides, you use ATP. So just to stay alive, you need to constantly be burning ATP in a cell. And if you're not remaking ATP, the concentration will steadily go down because you're not replacing what you use. And the way you make ATP inside of a cell is glycolysis and then oxidative phosphorylation. So if you slow down glycolysis, you slow down ATP production, which means that cell gets more glucose delivered to it. So yeah, it's a vicious cycle. All right, so if there's no other questions about that one, and hopefully it was helpful to actually take a step back and think about the big picture ideas of these pathways. Question 4 is just more of a remembering the structures. Which carbon number leaves glucose to become CO2? Which one was that again? I'll give you a hint. Is it carbon 1, 2, 3, 4, 5, 6? Not C2, not C6. Nope. Let's go back then. We forgot. The carbon that leaves to form CO2. Carbon numero uno. Carbon number 1. So 1, 2, 3, 4, 5, 6. It's the one that turns into a lactone. That's the carbon that's going to disappear. Yep, C1. And then another just little review. What molecule branches from glycolysis to be fed into the phosphate pentose pathway? And we just talked about this, so I'll just give it away. I just went on for like 10 minutes about that. Glyceraldehyde phosphate and fructose 6-phosphate and glucose 6. I gotta work on my G's and my 6's. Glucose 6-phosphate. So those are the 3 molecules that connect directly into the pentose phosphate pathway. Oh, I guess I had separate ones, 5 and 6. So my bad. It branches from those 3, but it only returns versus in GAP, glyceraldehyde phosphate and fructose 6-phosphate. We cannot return to glycolysis from glucose 6-phosphate because stage 1, remember stage 1, which is our redox reactions, which uses glucose 6-phosphate, irreversible. Once we use that, there is no going back. So we can never return the glycolysis via glucose 6-phosphate in the pentose phosphate pathway. But we can return, if we want, via GAP and F6P. And that's what this image is showing here. Here's a much more zoomed out version of what we've been talking about, where now we have a better picture of glycolysis that we can add this other pathway that's also connected to glycolysis. So here we have our glucose going to our glucose 6-phosphate, can go to ribulose 5-phosphate. Doing that, we make 2 NADPHs for every 1 molecule of G6P. Ribulose can make ribose, which makes nucleotides, or we can feed it back into glyceraldehyde phosphate and fructose 6-phosphate. What pathway is turned on when? Like, let me rephrase that. For a cell to determine which enzymes it wants to use at any given time, it really depends on what it needs, right? If we really need NADPH, we are going to return to glycolysis. The logic of that is that if we really need NADPH, that's going to be my black lines. We take our glucose 6-phosphate and we make it into ribulose and make our NADPH. Then we go to GAP and F6P. And when we're in fructose 6-phosphate, we can remake glucose 6-phosphate and rerun the cycle again. So by doing that, we are spending ATP to just recycle our carbons to get NADPH. So if the body needs NADPH, they're going to do that giant black circle I just drew out. If we want to make ribose 5-phosphate, which I'm going to use in this dark red, then we're kind of going to go the opposite way. Well, G6P is still going to make ribulose to make ribose, but our fructose 6-phosphate in our GAP will go and make ribose as well. So you can see, based on what we need, we can do different things with our metabolites. And the flux, it's going to be mainly controlled by glucose 6-phosphate dehydrogenase. Remember, this is enzyme 1 of stage 1. phosphate pentose pathway, PPP. This is what's converting our glucose 6-phosphate into our glucose lactone, glucose 6-phosphate lactone. And we mentioned this, but it is regulated by our NADP and NADPH concentrations. You can see delta G extremely negative. But if NADPH is around, reaction 1 is inhibited. However, when you use NADPH, you make NADP over again, this will go and turn on glucose 6-phosphate. Whoops, that's supposed to be NADPH, not NADH. That's NADP. So that is how really we can control what we're doing based on metabolites. And so this is under hormonal control. Another way, long-term way to do this, but a much slower way, is just you can create different amount of enzymes, by different hormones. So any questions about what's presented on this slide? All right, then we can move on then. All right, let's take another step back and just realize what we've looked at. We're going to cover all of this. So far, we have done this. So we've covered the middle portion. Again, this is just to show you how interconnected everything is, how glucose 6-phosphate, you can see there in the middle, glucose 6-phosphate and pyruvate are really the big points where you go into different pathways. Now, the next thing we're going to talk about is glycogen and how we can use glycogen to make glucose 6-phosphate. So we're going to talk about breakdown for the rest of today. And then next week, we're going to talk synthesis. And hopefully, when I say the word glycogen, that's at least ringing some bells from Biochem 1. It's okay if you don't actually remember exactly what it is, but hopefully you have a tangential idea of what glycogen is. But we'll just refresh it. So glycogen is our storage molecule for glucose in the body. And it's alpha 1,4 linked glucose. So hopefully you remember our linkages, since we had a lot of those on our tests in Biochem 1. So hopefully alpha 1,4 is familiar to you. And we have our alpha 1,6 branch points. So this is a branch molecule. And basically you have, glucoses, and then every 8 to 14 glucoses, you have a branch point. A nice image is shown here. All of those spheres are glucose. The purple, as pointed out, is where you have a branching event. Blue is your non-reducing end. Hopefully you remember from Biochem 1, non-reducing means no free anomeric carbon. And the anomeric carbon is the carbon that's responsible for forming a cyclic sugar. So if that's in a bond, your sugar cannot become linear again. A reducing end means you do have a free anomeric carbon. In glycogen, you can see we have a lot of non-reducing ends and only one reducing end. And the idea, and we talked about this, but the idea is that this is your glucose storage inside the cells. And the enzyme that works to break apart these glucoses only works from non-reducing ends. So the idea is I can take many glucose molecules off at a time if I have multiple enzymes all working from the non-reducing end. While if I had an enzyme that only worked from the reducing end, I could only take one glucose off at a time because there's only one reducing end. Right. So in these molecules, you have something like 120,000 glucose molecules stored. Your muscles, roughly 1 to 2% of the weight of muscles is made up from sugar. Liver, 10% is made up of sugar. And we'll see later on in this course, your liver is massively, massively important when it comes to glycolysis. So please treat your liver right. Don't drink too much. Don't eat too much sugar. Your liver is incredibly important for glycolysis. Yeah, and the last 2 points here just repeat what I've already said about non-reducing ends. Any questions about glycogen, our linkages here, any of those vocabs we said? All right. Glycogen is just a bunch of glucose together. Yep, that's 100% what it is. Why is there one reducing end again? Sure. So if you remember what an anomeric carbon is, an anomeric carbon, basically, one way to look at it is where one carbon is attached to two oxygens. In our cyclization reaction, I'm going to do it very simple. It's the carbon that gets attacked to form our ring structure. So in this very simple cartoon, that was our anomeric carbon. All right. If my anomeric carbon is not in a bond, I can go back to being in linear form. That is called a reducing end if that anomeric carbon's not in another bond. So here on our sugar, since we're alpha 1,4, the very last sugar of our molecule is going to be the only sugar that has reducing end. It's the only sugar whose anomeric carbon is not in a glycosidic bond. For all the other sugars, they are all connected via their anomeric carbon, either 1,4 normal linkage or 1,6 branch linkage. So out of the 120,000 glucose molecules, 119,999 have their anomeric carbons bound. So they're not non-reducing ends, but the anomeric carbon are bound. Exactly one molecule is reducing. So I should say non-reducing. Why that's good for us is that the enzyme that chops up glycogen and releases free glucose that we can use for metabolism, it only works on glucoses that are non-reducing or the non-reducing ends. So let's say that, you are lifting weights, right? And you are using a lot of ATP to lift something heavy in your muscle. The way you make ATP is glycolysis. And so to get glucose fast, what will happen is that all of the branch points that are non-reducing will be chopped off at once. So in this example, I'm releasing 1, 2, 3, 4, 5, 6, 7, 8. You're releasing 9 glucoses at a time for every time a reaction happens, more or less, because you have 9 non-reducing ends. If, on the other hand, your enzyme worked opposite for some reason, if my enzyme only worked from reducing ends, I could only release one glucose at a time because I only have one reducing end. So that's a long-winded way of kind of explaining it again. Hopefully that helped. I mean, why is there one reducing end? There's two reasons. The way it's bonded, that's one reason. Two, the physiological reason of how the enzyme works and why it works that way. All right. Anything else, or that help? Okay. All right. So what we're talking about is glycogenolysis, glycogen lysis. Hopefully you remember when we talked about hydrolysis, that's water breaking. Lysis means to break. So glycogen lysis basically. And to do this, to break down glycogen, you need three enzymes. The first is glycogen phosphorylase. All right. Now, a phosphorylase is the opposite of a kinase. A kinase adds a phosphate group. A phosphorylase removes a phosphate group. And again, it's kind of in the name, like LY, almost like lysis. Not quite, but close. So glycogen phosphorylase catalyzes phosphorylases, and you'll make glucose 1-phosphate. So what's happening here? Even though I just said it's the opposite of the kinase, because it usually is. I guess this is the exception that proves the rule. We're taking glycogen, which is made out of glucose. We're breaking that bond, and we're making glucose 1-phosphate. So here, we're actually adding a phosphate. So I apologize for probably just confusing you right there, what a phosphorylase does, but I was getting ahead of myself there. Phosphorylase usually does take off the phosphate, but here we're actually adding one. Here is the actual chemistry that I was just talking about. Glycogen plus phosphate equals glycogen minus one glucose plus glucose 1-phosphate. The interesting thing about this enzyme is that the glucose must be at least 5 glucose away from the branch point. Let me just go back to this picture on the previous slide. So redrawing it. So our glycogen more or less looks like this. So here's a branch point in purple. So here's 1, 2, 3, 4, 5. Only these 3 glucoses could be used by glycogen phosphorylase. Once you get 5 away from the branch point, you can no longer cut off glucoses. So that's what we mean when I say 5 units away. Let me get rid of the 1, 2, 3, 4, 5. So when we want to get the glucoses near the branch points, we need a separate enzyme, and that's just called glycogen debranching enzyme. Probably one of my favoritely named enzymes because no confusing words there. It removes branches from glycogen. Once you remove the branch, so what we're doing here by removing the branch, so let's say like this is a branch. What the branching enzyme does is that it kind of like chops this off and moves it to one of the ends of glycogen. Now our glycogen phosphorylase can work again because we're not at a branch point anymore. So it will just be completely linear. That's what I'm trying to show in that picture. Not that you make another branch, but it becomes linear. So glycogen phosphorylase can work again. The last enzyme you need is a phosphoglucomutase. The reason for this is to go into glycolysis, you need G6P. However, glycogen phosphorylase only makes G1P. And so this is a simple switch from G1P to G6P. Again, mutase, all a mutase does is that it switches these functional groups for us. It moves where the functional groups are located. That's what a mutase means. So any questions about that info? Okay. Just seeing how much we have to go. Okay. So let's take a look at this glycogen phosphorylase enzyme. And I'll rewrite what I have down here. And this will probably be the last slide for today. And then we'll just pick up the last few ones on Tuesday. So this says phosphorylase B. This says phosphorylase alpha. All right. So glycogen phosphorylase, which chops off glucose from our glycogen storage, has two forms, R and T. See, there is a reason why we studied hemoglobin so much in Biochem 1. It's because we use R and T for everything. R being the relaxed or active state, T being the tense state. All right, so let's take a look at the picture in the bottom right that's showing this. One thing to note, even this has like 4 boxes. This is just one enzyme. Each one of these boxes is a different state of one enzyme. So we're not looking at 4 enzymes, we're looking at one enzyme. The red, both of these reds here are 2 different forms of the T state. So T state can be either phosphorylated or unphosphorylated. So these P's are phosphorylated. Or we have the R state in the green, which is either unphosphorylated or phosphorylated. If there is no phosphate attached to our enzyme, we say it's in the beta form. So that's why it says phosphorylase beta or B. If we do have a phosphate attached to our enzyme, we call that the alpha form or phosphorylase alpha, right? So that's how this picture is oriented. So first, we are going to just look at the beta form, how we switch from the T to the R state, right? So this breaks down glycogen. We want to break down glycogen when we're low energy because glycogen is our fuel storage. So to go to the T, from the T to the R state for the beta enzyme, AMP will do that. Remember, AMP is the signal we're low energy. So for low energy, turn on this enzyme to break down glycogen. What will turn this off is ATP or glucose 6-phosphate, right? ATP says we're high energy, so don't break down our storage. Glucose 6-phosphate is, if there's glucose 6-phosphate around, that means don't break down our storage. We already have glucose ready to go. The thickness of these arrows are actually telling us something. If we're in the beta form of this enzyme, you are majorly going to be in the T form. So if you do not have a phosphate attached to this enzyme, you're going to be turned off, mostly. The exception is if you have AMP around, you can turn on this enzyme to the R form, but it most of the time it's off. To really turn on our glycogen phosphorylase, you need to add a phosphate. That's what a kinase does. The kinase, here phosphorylase kinase, because we're talking about glycogen phosphorylase, the kinase will add a phosphate to our molecule, to our enzyme. Once a phosphate is added, you more or less automatically go to the R state. That's what this arrow is showing, right? This arrow is thick and it's saying once you add that phosphate to this enzyme, you are in the R state. You are ready to break down glycogen. You can go back to the T state if you have glucose around. If you have a lot of glucose, you can turn off this enzyme. But for the most part, once you're in the alpha state, you're going to be on. So in the alpha state, the R form is preferred. So really, the way that this enzyme is regulated for the most part is the phosphate group. Is it phosphorylated or is it not phosphorylated? And we'll look later on in the semester. I know I keep saying that, but we're going to look later on how these two enzymes are regulated, how we turn on the kinase or how we turn on the phosphatase, because that is determining if we break down glycogen or not. Just some extra tidbits here. The molecule we're adding our phosphate is to a serine, serine 14. Hopefully you remember your side chains. Serine has an OH side chain, so that's where we're adding the phosphate. But yeah, the big takeaways here is, and what I would hope is, can you understand like this image? Like, can you explain this image back to me? And do you understand the logic of what's happening in this image? Do you understand the logic of why phosphorylase should be turned on or why it should be turned off? And remember, this breaks down glycogen. When you break down glycogen, you get glucose. When you go through glucose through glycolysis, you get ATP. So remember those concepts when looking at this image. Remember R is on, T is off, and try to remember all that. And I think the logic of this image will come through. Right. Any questions about what I said there or anything at all before I let you go for the week? All right. If not, hopefully you learned something today. Hopefully you got to see how cool it is to see everything interconnected. I will put a homework up as always. Reading guides will continue as usual on Tuesday. Our first test is on the 16th. So basically 3 class periods from now will be our test. And I'll talk more about our tests next week. Otherwise, if you have questions, you want extra tutoring sessions, anything like that, don't hesitate to contact me. Other than that, I don't have anything for you. Thank you for stopping by, and hopefully I'll see you next week. Have a good one, everybody.